Today it is my pleasure to host and introduce Dr. Roxanne Banker. Roxanne is currently a postdoctoral researcher in my lab in IZ&G. She joined us in January. I'll give her a brief little introduction here. Roxanne has an undergraduate degree in biology from Denison University in Ohio, which I believe he completed in 2013. And then she moved to UC Davis, a place near and dear to my heart, where she did her PhD in the Department of Earth and Planetary Sciences. And interestingly, even though I'm Roxanne's postdoc mentor, we are also academic siblings because she did her dissertation work with my PhD advisor, Yurat Vermey, Academy stalwart and fellow. Roxanne's PhD work, her thesis work, I think reflects her transdisciplinary skills and interests. In her background from both her undergraduate and her graduate work, her research experience and her interests in general span geology, biology, stable isotope geochemistry, ecology, and even microbial genomics. So currently in my lab, she is funded by an NSF grant. She's working on two projects though. One, the one that she was actually hired to work on, looking at ancient terrestrial ecosystems in northwest China. We're not actively looking in China these days, but we're still working on it. And she's also working on a project, a heavily collections-based project, which we hope to get off the ground soon, looking at the evolution of modern and shallow fossil record marine ecosystems in the Northeast Pacific. After finishing her PhD at Davis, she did a short stint as a postdoc in the Department of Earth and Planetary Sciences with David Gold, who's a relatively new arrival there, fresh out of Caltech. So I'll turn it over to Roxanne, and the title of her talk today reflects her interests, I think, in oysters and shells and omics. Oh my. Before we get started, actually, two quick reminders. One, if you're listening in, please be sure to mute your mic. And if you would like to ask a question, pop it into the chat box and prefix it with a Q, and I'll try to get to as many of the questions as we can. Okay, go for it, Roxanne.
Great, thank you so much for the introduction. And so like Peter said, I'll be talking a little about some of my previous work. So this will touch on one of the chapters in my dissertation and also that brief stint as a postdoc I did with David Gold. So today I'll be talking about, from my dissertation, the role of sulfate-reducing bacteria in oyster chalk formation. And then sort of the second part is that postdoc work that I did with David Gold. So I called that larval to juvenile oyster shell development, a multi-omics approach. So just a little bit of background, specifically for my dissertation, I came in and I was really interested, or my project is focused on better understanding this unique shell feature that some oysters produce known as chalk or chalky deposits. And on the right, you can see a figure modified from McDonald et al. 2009. On the left is an image of the shell folia, which is what the majority of the shell is composed of. So you can see that the crystals are really tightly packed together and there's not a lot of void space. On the right-hand side is an SEM photo of chalk, and both features are calcite, but the chalk crystals are arranged in a way to accommodate a lot more void space. And so that feature, if you interact with an oyster shell, it's really soft and it's a lot less dense than the rest of the shell. And so I was interested in this because a lot of people had talked about it over the years in various papers, but the ecological function was highly debated, as was the mechanism of formation. So some people had thought that it was primarily formed by the animal itself. Others hypothesized that because of the unusual microstructure that there might be some microbial involvement in precipitation. And so I was just interested in better understanding this trait to understand the modern ecology of organisms. So what do they use this feature for? Why is it important? But also try to better understand the evolutionary history of that trait and the oyster shells. And so really the guiding question for this was, are chalky deposits in oysters caused by bacterial metabolism, specifically bacterial sulfate reduction? And the reason that sulfate reducers are of interest here is because sulfate reducing bacteria are sort of the major players in carbonate precipitation and microbial mass. And this is, and they do this by a few different ways. Their bacterial sulfate reduction might increase the pH, increase alkalinity, or alter organic compounds to create nucleation sites. And just to get us all oriented for anyone who's not very familiar with oysters. So on the left is an internal view of a left valve. And you can actually see the chalky deposits are kind of conspicuous. They're these opaque layers. Here's the umbo and the commissure, which represents the growing margin. Here's the cross section through a shell. It's actually not from this one, but just to show you what it might look like. The translucent gray layers are that foliated material. And then these white lenses are chalk lenses. So you might even say they're chock full of calcite. So the first thing I wanted to talk about today was, for my dissertation, I did some experimental manipulation of sulfate-reducing bacteria in oysters in order to see how this might affect chalk reduction in the shell. So the species that I used for this work was the Pacific oyster. These are farmed extensively on the coast of California, so they're easy to obtain. And they also grow really quickly, which makes them ideal for a wide variety of experiments. So the individuals that I obtained were from Starboard Mariculture, and I built these mesh cages to house them. They were outplanted. I tied those cages to a dock in Bodega Harbor, so they got a lot of nice free food. And then they were treated monthly to conditions intended to alter their internal populations of sulfate-reducing bacteria. So I ran that experiment for about 7 months and sacrificed them in January 2017. So here's a view of their outplant location. This is a commercial upweller system that's owned by Starboard Mariculture. And so my oyster cages would have been tied to the dock right near there. So you can just see it, there's a wheel turning, and that brings all of the oysters that they have on this raft a lot of food, and it brought mine a lot of food. They grew very quickly. On monthly treatment dates, I brought the oysters from the cages to the marine lab, where I rinsed them, cleaned them, and then I put each treatment group in a bucket with a healthy serving of algae to eat, incubated them in treatment reagents for about 3 hours, and then rinsed them and outplanted them back in the harbor. So the treatment conditions are as follows. First, of course, we always need a control. So those oysters were moved from the cage to a bucket with no reagent added. They just had algae with filtered seawater. So that should be our sort of baseline, no change in the abundance of sulfate-reducing bacteria. The second treatment, I wanted to see if I could knock down internal populations of sulfate-reducing bacteria. So I exposed the second group to ciprofloxacin, a broad-spectrum antibiotic. Based on a paper that I saw by Cordova and Krelos and SCO 2007, they tested, they exposed salt marsh sediment microbial communities to Cipro and found that they actually saw an increase in sulfate reducers, and they hypothesized that this is because it knocked down their competitors. So I wasn't sure how that antibiotic would affect those populations. So I also added a third treatment where I added sodium molybdate to buckets. And this compound is known to inhibit bacterial sulfate reduction. Then the final treatment was I wanted to inoculate oysters with a sulfate-reducing bacterium. So with collaborators from David Coyle from Jonathan Eisen's lab at UC Davis, we isolated this disulfovibrio from a seagrass root that was collected from Bodega Bay. And I grew up liquid cultures, and that's what I used to inoculate the oysters with. So at the end of the grow-up period, about 7 months, I collected 20 of the 20 largest oysters from each treatment group. And I then assessed the abundance of sulfate reducers using a test kit. And I applied that to 10 oysters from each treatment group and 4 seawater samples as a point of comparison. Shell chalk content was also estimated by measuring bulk shell density. So again, chalk is less dense than the rest of the shell. So enhanced chalk expression would lead to a lower shell density. And then I also calculated chalk as a percentage of the cross-sectional area in the shells that I collected. So the way these test kits work is I shucked an oyster open the way you would when you eat it. I applied an applicator, these basically little pipe cleaners, to oyster fluids and seawater samples. And then you insert that applicator back in these test tubes and you top it with a mineral solution and incubate it according to manufacturer instructions. So then as the sulfate reducers are metabolizing, the sulfide will bind with metals in the solution, and the amount of black sulfide produced over a certain period of time is proportional to the abundance of sulfate reducers in the original sample. So here's what some samples look like before being incubated and what some looked like after incubation. And then based on the manufacturer protocol, you can estimate the number of sulfate reducers per milliliter based on this table. Oh, that figure is blurry. That's too bad. Hopefully it's not blurry for everyone else. So the way that I tested this data statistically was I went through each sample from all the oysters in those tubes and assessed their appearance, so how much of that black sulfate was produced, and then assigned them these values based on the manufacturer's table. So then I gave the lowest quantity a rank of 1, and then ranked each successively higher bin with a higher rank, all the way up to the highest quantity for a given sample, which received a rank of 7. I then plotted this data as a frequency distribution for each group, and then compared those statistically using the Kruskal-Wallis test. And so that yielded a significant result, and post-hoc pairwise comparisons yielded the following, or I like to present it in a Venn diagram. But essentially the takeaway here is that based on those statistical procedures, in terms of sulfate reducer abundance, the group exposed to disulfovibrio, the control, and the ciprofloxacin group were statistically similar in terms of their abundances. And then group 2 included the molybdate group, ciprofloxacin, and the control were statistically similar. Seawater had very, very few, or it wasn't really detectable number of sulfate reducers, so they're in their own group. But the important takeaway is that the disulfovibrio group had statistically more, or significantly more, sulfate reducing bacteria than the molybdate group. And again, that compound is designed to inhibit sulfate reduction. So that makes sense, which is good. And then the way I wanted a way to compare sulfate reducer abundance to the chalk in the shell. And so the 2 ways I did that were by measuring bulk shell density. So I took the dry weight divided by the volume of the 20 shells that I collected for each treatment group, and volume was measured as water displacement. And then the other way I did this is I had those 20 shells cross-sectioned, and here's one example of what that would look like. Again, here's the umbo and here's the commissure. And in image J, you're able to highlight the whole surface and then just the chalk and calculate those areas and get a percentage from that. So here are the results for shell density. Oh, this is supposed to be an arrow pointing towards the right. I don't know if that's showing up for people. But here, lower density is equivalent to having more chalk in the shell. So on the x-axis, there is treatment groups, so ciprofloxacin, control, molybdate, and the inoculum. And then on the y-axis is density in grams per centimeter cubed. And so the important takeaway here is that the control group has a lower density than all the other treatment groups, which are statistically similar in terms of their density. So basically, the control group produced more chalk when compared to the other treatment groups, and this was statistically supported. Similar results for the percent chalk measurements. So a similar graph where treatment is on the x-axis, percentage of chalk is on the y-axis. Again, the control has more chalk expressed in the shell, but this is not a statistically supported pattern for these data. I think that there might be some errors in the measurement and maybe would have affected the significance of this test, but I think that this is a real pattern. So overall, treatments were effective at altering internal populations of sulfate-reducing bacteria, but sulfate-reducing bacterial abundance did not correlate with chalk formation. So for example, the control group had about a medium amount of sulfate-reducing bacteria, but had the highest chalk expression. In contrast, the groups exposed to molybdate and the inoculum had different amounts of sulfate-reducing bacteria, but produced a similar amount of chalk. So what this indicates to me is that sulfate-reducing bacteria, as has been hypothesized, are not solely responsible for inducing this chalky feature in oysters. But I do think that the microbiome plays an important role in healthy organism function, including calcification. Unfortunately, the test kits here don't provide a lot of other data on this system, so sort of teasing apart other conclusions isn't really possible with this data set, but would definitely be worth looking into. Okay. And so this eventually, or I was finishing up this work as David Gold came to UC Davis, and we developed this other project to sort of look at the interaction of pH stressors and how that affects the microbiome and the gene expression of the oyster and how this might affect shell formation, again, in the Pacific oyster. So this was a little bit exploratory, but I'll talk a little bit more about that in a minute. So some of the motivation for this study is, I'm sure most, if not all of us, are aware that ocean acidification is an increasing problem, which is a result of, as we're putting more and more CO2 in the atmosphere, it diffuses into seawater, thus decreasing the pH. This reduces carbonate saturation state in seawater and can have negative effects for marine calcifiers. One example are oysters, and they're interesting because they precipitate both calcite and aragonite in their shell. And they're also ecologically important organisms because they filter feed so extensively that their activity can be important for water quality, especially in systems that have experienced eutrophication. And they're also a globally commercially important food source. And these critters have already had some, there have already been some mass mortality events for aquaculture oyster growers. So trying to better understand what factors are at play during low pH events that affect their mortality is important. So some of the questions I had going into this were, how do stressors such as pH affect the host microbiome, host gene expression, and shell formation? And we already have a few pieces of puzzles, but having that whole data set from one experiment, I think, will be helpful. And then if shell formation is negatively affected by lower pH, are there corresponding changes in the microbiome or gene expression that can explain this? But if shell formation is not negatively affected, are there bacterial taxa or genes that conferred resistance? So this was the basic experimental design, and this is a figure created by David. So what we wanted to do is grow oysters under 3 conditions. Normal pH conditions as a control, acidified pH conditions as an analog for ocean acidification. And then we also added a third treatment where we added, again, sodium molybdate, which is known to inhibit bacterial sulfate reduction. So even though those bacteria aren't responsible for chalk, they still might be important for carbon cycling or the carbonate system. Just a little more background on oyster development. So oysters begin their life as a planktonic larvae, and they spend about a month in that state, where they eventually settle and lose their ability to swim, and they settle to the seafloor on a hard substrate and grow there for the rest of their adult life. So here are some of those life stages. The trophophore larvae occurs very early on in the life cycle. After a few days, they make it to this D-hinge veliger stage, and they look like a D, that's the name, before they grow rapidly in size and reach this eyed petaveliger stage, which is when settlement occurs. It's right after that. And then here's what a juvenile or adult would look like. So this project was kind of fun because I got to actually spawn oysters with the with a lot of help from the folks at the Bodega Marine Lab in Bodega Bay. So again, we used the Pacific oyster, which is a fast-growing common aquaculture product. We used a process called strip spawning. So we had adult broodstock oysters supplied to us that were living in this tank in the background here. And then what happens is you shuck the oyster and you actually score the gonad and remove the gametes, stripping them. And we combined gametes from 2 males and 2 females in one of these big 1,000-liter tanks. And then larvae were allowed to reach the D-hinge veliger stage after about 2 days. And that's when they have a shell that's fully enveloping all of their soft tissues, and they're a lot less sensitive to environmental changes. So at this point, we transferred them to the treatment buckets where they would live for the rest of the experiment. And this is also when we took samples that would eventually be, I would extract RNA and DNA from them for the microbiome. But I'll talk about that in just a minute. Here's just another picture of that experimental design. So here's our big pooled larvae tank. And then after they reach the D-hinge veliger stage, we split them into those 3 treatment groups that I talked about. So the control, low pH, and sodium molybdate. And then there were 3 replicate buckets for each treatment. So they all started from the same sort of pooled source, and then were evenly distributed after they reached the D-hinge stage. And then here's what that actually looks like. So in the lab, all of those buckets are set up on a table here. The way that I controlled pH is for the low pH buckets, they were attached to these pH controllers. So there was a probe going into the bucket that ran to the controller, and then that ran to a solenoid activated valve on a CO2 tank. So when the pH got above a certain point, it would energize the valve and release CO2 into the system to drive it down. The control just had, all buckets also had an airline coming in just to keep oxygen high, and then the molybdate bucket had sodium molybdate added. So I also spent some time monitoring the seawater conditions while this experiment was running from about, August 28th to mid-October. I should have written that down earlier. But so each day I would record temperature, pH on the probe, dissolved oxygen using a monitor or a probe, and salinity for each bucket. Because these are static cultures, they're not in a flow-through system where the water is being recycled. I also had to change the water every day. So that involved taking each bucket, pouring the larvae onto a sieve so they didn't get lost or go down the drain, rinsing the larvae into a clean bucket, and then adding fresh seawater. And so during each water change, I took a sample of seawater to be analyzed on a spectrophotometer shown here for a more accurate measure of pH. And I also took a paired water sample to analyze alkalinity. Oh, and here's what one of those seeds look like. So these small grains of sand that you see are actually like millions of small oyster larvae. Right. So the way that I collected samples for RNA extraction and for the microbiome analysis is from that group larval tank, we poured a bunch of water onto a sieve, and then I rinsed a small spatula with ethanol, took small scoops of the larvae, put them in a microcentrifuge tube, and froze that immediately. Let's see. So then we allowed the oysters to grow out until they settled as spat and became juveniles. And I used a pretty similar procedure to take samples of the juveniles, where they had already settled and permanently attached themselves to the bucket. So I actually had to scrape them off, get them in a tube, and harvest them that way. And then importantly, there are 2 juvenile time points. So I destructively sampled buckets 1 and 2 for each treatment 12 days post-settlement. So after that point, we only had 1 replicate for each treatment running. And then I sampled those 25 days post-settlement for each treatment. Here is what some of the data looks like. So seawater pH on the x-axis is day post-fertilization. So day 0 was the day that we mixed the gametes together and the eggs were fertilized. Day 2, about right here, would have been, day 2 is when we transferred them in buckets, and then day 5, when we get the first data point, is when I did the first water change. And then on the y-axis is pH. These bars represent the duration of the replicate. So replicates 1 and 2, I sampled from at day 12 days post-fertilization, so about day 38. And then after that, the numbers are only represented by 1 bucket. So the control and the molybdate replicates all stayed at about a pH of 8. And I should have added the numbers here, but the average is about 8 for all 3 replicates from the control and the molybdate groups. The pH system was not perfect. Obviously, these are pretty significant swings, but the average pH for these low pH buckets was about 7.7. So no need to memorize these numbers, but this is just my slide for talking a little bit about sequencing and bioinformatics. The DNA from oysters was extracted using a Zymo kit, and then that material was submitted to Dalhousie University for 16S rRNA, PCR, and sequencing. I used the DATA2 pipeline in R, and that yielded, these are the, let's see. So these are the bucket IDs on the left, and then each column is the number of sequence reads that came through various filtering and denoising steps in the bioinformatics pipeline. So then these are the final numbers of reads, which is represented by almost 2,000 taxa across 38 samples. So let's see. So here is a summary of the alpha diversity from those different treatment groups. On the x-axis are the bucket IDs. On the left shows the Simpson index, and on the right the Shannon index, just to compare the two. Each bucket, so bucket C1 is represented by two replicates, and this is from the microbiome data. So these diversity measures were calculated based on the taxa from the bioinformatics pipeline that were found in each treatment. So for the most part, based on the Simpson and the Shin index, all the sample buckets seem to have similar diversity. I actually haven't run statistics on this yet. You'll notice that the bars, and the bars are one standard error. And they're very, very big for the sodium molybdate second replicate bucket, SM2. And that's because one of the samples here ended up falling out of sequencing. I think, yeah, SM1 is only represented by 2 points because the third one fell completely out. And so that's why its error bars are a little nicer. Okay, so next what I did is I, based on the community composition from each of those buckets, I used programs in R to calculate a Bray-Curtis distance and then plotted this in a principal coordinates analysis space. So basically what you're looking at here is an ordination where the distance between points represents their compositional or their dissimilarity. So points that are closer together are more similar compositionally, and points that are further away are more different compositionally. And so here are the larval samples on the left, and those are very different from everything else. So this is still work in progress, so I'm not sure which taxa are driving that difference yet, but I think one of the major factors driving this distance is that the larval samples had about an order of magnitude fewer reads than all of these. So I think that's part of the story there. Oh yeah, larval samples. Next, I removed the larval samples because they were sort of compressing the graph from the ordination, and that yielded this plot. And so what I thought were a few interesting patterns were, so first, if we look at just the control group samples, C1, if you remember, C1 and C2 were collected at the same time. C3 was allowed to grow out for another 15 days or so. And so C1 and C2 cluster really nicely together. And then C3 is different than that. And I'm not showing it here, but this is statistically supported. I ran a permutational ANOVA in R, which yielded a significant result. Next, and you can see the same pattern across all the treatment groups. So it seems like there is some community composition shift that occurs from here from 12 days post-settlement to 25 days post-settlement. And again, I'm not really sure which taxa are driving that difference at this point. I haven't gotten a chance to run those analyses, but I think that'll be very interesting. And then the same pattern is found again in the low pH data as defined by these OA samples. So these are those sampled 12 days post-settlement and 25 days post-settlement. Last, I also plan to make a nicer plot of this, but here's just a taxonomic bar chart showing I removed the top 20 most abundant taxa from each sample bucket and then plotted those here. So there's this C3 sample that looks a lot different from all the others, which could be contamination or it could be an interesting signal, but again, that's something that I'll need to dig into a little bit more. So those are the analyses that I've run on the data so far. As I'd indicated before, my next steps are to determine which taxa are driving differences between treatment groups, because those, I didn't mention this explicitly, but the different treatment groups are also statistically different in terms of their composition. So I want to find out what taxa are driving the differences between groups, what taxa are driving the differences within a group between those two time points. One thing that I didn't mention is that for all of these microbiome samples, we have paired samples where I took the oysters and extracted the RNA. So eventually we'll be able to get gene profiles for the oysters themselves, but I submitted that data earlier in January, so I think it's stuck in limbo because UC Davis labs are shut down right now, but we'll see. And then I also need to analyze the shell data. So I took SEM photos of about 20 shells from each treatment group. And I plan to take measurements of the shells themselves. But what's cool about these oysters is that you can actually really clearly see the transition from their larval shell to their juvenile shell. So I'll be able to get some information out of comparing the ratio of the larval shell to the juvenile shell. And then I'll be able to assess if the different treatments had an effect on shell formation and tie that back to the microbiome data. Acknowledgements, and that's it. That was a little bit short, but it's digital, so yeah. Any questions?
Do we have any questions, Roxanne? Remember to type them into the chat box. I actually have a couple of questions, Roxanne. First, just a general one for the listening audience. Could you pop in a quick explanation of aragonite and calcite?
Oh, yes. So, yeah, I tossed that in there, but didn't explain it much. So aragonite and calcite are both forms of calcium carbonate, but their atomic structure is arranged differently, so it confers different physical properties. Aragonite is generally less stable than calcite.
Yes,
Yeah.
Go ahead, sorry.
Oh, and then the other thing I was going to say is that these oysters are interesting because they form an aragonite shell as larvae, and then they immediately start precipitating mostly calcite after they settle. So I'm really excited for the RNA information to look at which genes are being used during those two different time points. I forgot to mention that. Thank you.
Yeah, that's cool. I don't think we know anything about that actually, but do you have any thoughts then on the impact of acidification on the different life stages, given that they go through such a dramatic shift in their structure?
Referring to the change in mineralogy?
Yeah.
Yeah, well, honestly, I was really surprised during this experiment that, the reason I had so many replicates is because I was expecting some mass mortality events, especially in the low pH treatment, which I didn't see. So I think normally that's a really sensitive transition and where a lot of mortality does happen in aquaculture, I believe. So as for predictions, I think they've already been sort of shattered by the survival rate of my experimental organisms. So I would have expected them to do more poorly. And just looking at the shells, I haven't analyzed them yet, but just looking at them, they didn't look smaller or worse. So I guess I had expectations and they have not been met. Yeah.
Okay, thanks. And then one other quick question. With such a shift in the microbiome composition, is that an inoculation that's taking place post settlement, or how do you think that's happening, especially into your lab conditions?
Yeah, so you're talking about between the 12-day and 25-day critters? Yeah. So why would there be such a shift? Yeah, I've thought about that. It could be, it is possible that the incoming seawater from when there was some big microbial shift in the incoming seawater that occurred over that period of time is possible. But I would think that as they're growing and, I think we started feeding them maybe a different kind of algae, so that definitely could have contributed to it. So yeah, it could be their diet, or maybe they're attracting different microbes at different life stages to help them assimilate nutrients differently. I'm not totally sure. As far as I know, no one's answered that, but those would be my first guesses. If there is a shift in the seawater or the diet, or if they're attracting things differently over their life cycle. Yeah. Does that answer your question?
Yeah, it does actually. And if there are no other questions coming up, aha, we have a question from Shannon, but I have a follow-up. Okay, so Shannon says to follow up on my question. In some cases, microbial distance is greater over time within treatments versus between treatments.
Oh, okay. Yeah, so I'm not sure, I guess would be my answer up front. It does look like it based on the PCOA and some of the statistics. But I guess I could also look at that another way. But yeah, that is what it looks like in some cases, and I was surprised by that. And that's not something, right? Yeah, elaborate. Triangles versus circles. Yeah, so one thing that I thought was really interesting, is so there's like the 12-day control samples and the 12-day molybdate samples, and then their 25-day post-settlements are really similar. I was just looking at that today, and I don't know what to make of that at this point. But I am planning in the next few days basically to run a few additional analyses that should tell me which taxa are under or overrepresented, and then maybe I can see also where there's a lot of overlap between those circles and triangles. Yeah, hope that answers your question, but I don't know the answer yet. So Darrell asks, in your initial chalk study for the bar chart part, you didn't find a significant result with your statistics, but there was a possibility that of a nonlinear response hidden in the data, a peaked distribution. Nonlinear response. Honestly, yeah, I mean, it seems possible. I don't know exactly. Yeah, I think, yes. I don't know exactly how to tease that out, but that does seem to be the case, yes. I hope that answers your question.
Could you pull up the figure maybe, Roxanne?
Oh, yeah, sorry.
No, that's okay. Different format.
And this is it, right? Yeah. Is this the one you were talking about, Darrell? I actually can't view the chat on my slides at this time.
Yes, that's one.
Okay.
I think I lost track of what led up to that, but yeah, we can talk later. I think it was super fascinating. Okay. Thanks a lot.
Yeah, I would be happy to talk about it. Mm-hmm.
Good job.
Some sort of threshold effect. And then, are there any more questions? Let me check. Okay, then I have one final one, okay. A broad one, Roxanne. We have some other bivalve groups like lucinids, for example, that have symbiotic, might be obligate, sulfate-reducing bacteria in them. They have for a very long time, geologically, evolutionarily speaking. Any sense as to how those might interact under changing pH conditions? I mean, we think we sort of know why the bivalves have them, as we live in these environments. Yeah.
Yeah, because the sulfur oxidizers and synods help to deal with the sulfides that get entrained in seagrass sediments. Is that
Yeah.
what I'm thinking of? So, yeah, I'm not exactly sure how it would change, but I think, it depends, I think, a lot on how seagrasses respond because, or how the environments in which they live respond. I don't know if I have a clear answer for this, but I guess part of the ocean acidification project that I was doing with David is looking at the microbiome, and I think it's underappreciated how important different microbes are to taxa, and it's also not well understood how those microbial communities will be affected by climate change. But people have definitely already observed, and like I have, like shifts in the microbial community, during pH change, during temperature change. So it might be like corals where under some stress, they don't interact with their symbionts as effectively. So that might affect the leucinid symbiosis. But I'm not sure precisely what the effect might be.
Yeah. I wonder if the seagrass folks at Bodega looked into the microbiomes at all.
Yeah, a friend of mine, Cassie Ettinger, working with Jonathan Eisen, has done a lot of work with the seagrass microbiome, specifically looking at, well, she's done bacteria, but she's also focused a lot on the mycobiome, or like the fungi associated with different parts of the plants. So she's definitely looked into that.
Cool, thank you. And Darrell had a follow-up comment. He says, oh yes, it looks like a treatment overall effect. Everything did something to chalk compared to the controls. Right.
Yeah.
Could you tell about how treating the oysters did something, example, affected overall health that reduced chalk?
Yeah, that was something I was thinking of is, did the way that I disturbed the oysters like affect their overall health so they weren't growing as quickly, or did it affect shell formation directly? I'm not sure, but it did one of those things at least. I think I might have another figure actually where I have just shell weight. Yes, Cassie. But yeah, so I'll look into that though. I think I might have another figure that might explain that better, but I've lost track of it.
Okay, well, if there are no further questions, I'd like to thank Roxanne for presenting this week's seminar. And thank you everybody for attending virtually. I hope everybody is well, and we'll see you all again, I guess, same time next week.
Great. Thank you, Peter, and thank you so much for everyone. Yeah.
Thanks, Roxanne.